Hello everyone, this is an introduction to the new course in NMR spectroscopy which is titled as Multidimensional NMR Spectroscopy for Structural Studies of Biomolecules. In this course we will build on what the basics which we have learned in the previous course and we will go beyond 1 dimension and 2 dimension NMR and look at 3D NMR and its applications to studying structures of biomolecules such as proteins and nucleic acids. As you can see here in this slide, NMR has a variety of applications in different areas of science such as soil science, agriculture, medicines, in food technology and so on. What we are going to focus on in this particular course is this part which is structural biology, which concerns the structures of proteins and nucleic acids and their interactions with other molecules, small molecules. So, this course is basically going to benefit those who are working in the area of protein structures, working in the area of drug design, drug discovery, and those who are interested to study the interaction of small molecules with larger systems, larger biomolecular systems. So, NMR has a rich history in science. You can see there are 4 Nobel Prizes been given in one particular field which is unique to NMR, this is starting from the very first discovery of NMR phenomenon by Bloch and Purcell. Richard Ernst got Nobel Prize for 2D NMR and developing FT NMR technique, which is what we are using nowadays. Kurt Wuthrich got Nobel Prize for development of NMR for biomolecular structures, and this is going to be the focus of this particular course, where we will see how NMR techniques have grown in the recent years, and they are fascinating techniques, very nice approaches which can tackle challenging systems, which is not possible even by any other techniques, even including X-ray crystallography or cryo-electron microscopy. So, this is how the structure of the course will be. We will start from first few weeks will be very basic principles of NMR spectroscopy. We will look at just the principles of 1D and 2D NMR experiments, a few examples. Then we will move on to the 3D NMR part, which is the new part in this course, and here we will see what is a 3D NMR experiment, how is it made, how is it constructed, how it can be implemented, how it can be used. Then the second half of the course, then we will move on to the structures of proteins and nucleic acid. We will see a basic introduction to their structure, what are the special features. And an important thing in NMR for proteins is what is called isotope labeling. We do not work with just proton as in a 1D NMR. Here we need to have carbon 13 and N15 incorporated into the molecule. For that we need to use a technique known as isotope labeling. This is something which involves a lot of biology, and we will look at that. And in the last part of the course we will basically focus on how to determine structures, having isotope labeled them, and if we use 3D NMR and 2D NMR, how we can look at them, how we can look at the structures at high resolution. And also we will look at some applications of NMR for studying protein ligand. Essentially NMR is the very most important technique when it comes to studying protein ligand, and that is what is done in drug discovery. So, this is essentially the week 1 to 4 and week 5 to 8 topics. So, that is the first half, when the first half will cover the theory and principles, and second half will be applications. So, these are the following books which I would suggest, which can be used and referred to during the course. A few of them are listed here. Of course, the lecture will incorporate notes from many more books apart from what is listed, but these are very nice important books and very basic books. So, I would suggest to go through this. So, I look forward to seeing you in this course. Thank you.